students. Let's complete our talking about DNA. Let's complete chapter 8. In chapter 8.2, we're gonna talk about the structure of the DNA. In the previous lesson, we were talking about the identifying of the DNA as the genetic material. As I told you, the scientists did many experiments and many analyses for the genetic material to make sure that the DNA is the genetic material. After identifying the DNA as the genetic material, it was important for scientists to know the structure of the DNA. Knowing the structure of the DNA means to know the chemical structure and even the 3D structure of the DNA. Then they started to study the structure of the DNA. First of all, if I asked you, do you think the DNA is a monomer or a polymer? Do you know first what the meaning of monomer? Monomer is a small molecule or small subunit that can join two others to form longer chain. Look at this small ball. This is the monomer. One unit that can, or one subunit that can connect two others to form polymer. So, which one is larger? Polymer is made up of monomers. Very good. So, the monomer is the smaller subunit. The polymer is the long chain that is made up of many monomers connected together but by different bonds. Okay, do you think the DNA is monomer or polymer? Actually, the DNA is a polymer. So, if the DNA is the polymer, what do you think the monomer is? The monomer is nucleotide. So, the nucleotide is this small piece or this subunit, monomer, nucleotide. Many nucleotides join together the DNA. So, if the monomer is nucleotide, the polymer is the DNA. To study the structure of the DNA, of course, it's important to know the composition of the monomer. Since the DNA is composed of many nucleotides that are joined together, it's very important to know the structure of the monomer, which is the nucleotide. Actually, there are four types of nucleotides. So of course, there are four different nucleotides. So, can I tell you, or can I know a general structure for nucleotide? Yes, we can know the general structure, and after that we will know the difference between those four types. I told you the DNA is a long chain of nucleotides, and the DNA is the polymer, and the nucleotide is the monomer. Let's know the structure of the nucleotide. Do you remember when we were talking about Charles and Chase experiment? We said that they identified the DNA by tagging the DNA with, do you remember? Radioactive phosphorus. What does it mean? It means that phosphorus is one of the atoms in the nucleotide. Exactly. They found that each nucleotide composed basically of number 1, phosphate group. Here is the phosphate group. Number 2, sugar, which is called deoxyribose sugar. This is the deoxyribose sugar. Moreover, it contains nitrogen containing base. Okay, but you told me there are 4 types of nucleotides. So I didn't find any difference. Actually, the difference in the type of nitrogenous base, because they found that there are 4 types of nitrogenous bases. Number 1 is called thymine, 2, cytosine, adenine, guanine. Okay. If I asked you, are they similar? You will tell me, of course, different. They are different. The difference in what? Look at the rings. Are the rings similar? No, there are different substances in the rings. But I have two bases that have single ring. Look at thymine, cytosine. Thymine has only one ring. Cytosine has also only one ring. And I have another two bases that have two rings, double ring. Look, double rings. 1, 2. And also guanine. So if I asked you, in what way thymine and cytosine are similar? They have only one ring. In what way adenine and guanine similar? They have double rings. The nitrogenous bases that have double rings are called purines. Nitrogenous bases that have single rings are called pyrimidines. Okay? So now we know the structure of the nucleotide.